Hey guys, back to some DNA to RNA to protein discussion here. Now, in this lesson, we're going to take a look at RNA. RNA is one of those molecules that has to go in between. DNA is your master blueprint. You don't want to screw that up. So, what we do is we make a copy of RNA, and that is one of those pieces of information that if it gets run down or destroyed, it's not a big deal because we can always make more copies. So, we're going to take a look at what some of the differences in RNA are and the different types of RNA that are used, and then we'll get into just a little bit of how RNA is made. So here we go. Now, this is part of a bigger process here. What we're going to be discussing is the process of transcription first, and that's where we're actually making the RNA out of the DNA. This happens in the nucleus because the DNA can't leave the nucleus. The DNA is stuck there. The RNA eventually can leave. It goes out into the cytoplasm and it goes to those ribosomes. And you can see that ribosome down in the lower right-hand corner here. This is just a simple model of the second part of this called translation. And we'll get into that a little bit more later. But if you put these two pieces together, this is what we get into what I call central dogma. Now, don't laugh at the diagram. I know it's kind of kindergartenly, but I just wanted to give you some ideas to how this process works. Now you can see the DNA in the dark blue here. The DNA is double-stranded, and that's the master copy. Now you can see that this section right here has been opened up. We have to open a book before we can read it, so we've got to open DNA up before it can be read. And you can only read one page at a time. You can only read one strand of DNA at the time. So this is the strand of DNA that's being coded into RNA. This pink part here is a special enzyme we'll talk about a little bit later called RNA polymerase. And then we're going to make this massive strand of mRNA. That's one of the three types. Now, before we can use it, there's a little bit of garbage in here we need to get rid of. So we trim those out, make it a functional piece of mRNA where it goes out of the nucleus. This right here is the nuclear membrane with a pore in it. And it exits, meets up with a ribosome, and that's where we'll start to make the proteins. So that's just an overall look at things. We'll take a look at some of the specifics here, and we'll break this down into some simpler pieces. So here are some basic differences between DNA and RNA, and we've got a model of both of them side by side. Now, first one. DNA gets its name deoxyribonucleic acid from the sugar deoxyribose. In RNA, we have the same 5-carbon sugar. It's just not missing one of those oxygens, so we call it ribose. So one of the key differences here is the sugar that goes into making them. The second one is that RNA is single-stranded. DNA is double-stranded. That makes it easier to work with. Now, the reason that it's single-stranded is it's going to be read right away, and it's going to carry that message very quickly. But in order to make it single-stranded, we've got to make one more change. That last change comes in this base called uracil. Notice that the 3 nitrogenous bases, guanine, cytosine, and adenine, still exist, but you only find thymine in DNA. In RNA, you have uracil. That still pairs up with adenine. So when we looked at those base pairing rules in DNA, A bonds to T, G bonds to C, unless you're talking about RNA. If you're making an RNA you're always going to have a U opposite of an A. Okay? Now we're going to take a look at 3 different types of RNA. The one that we're going to have our primary focus on is something called messenger RNA. That's going to be the working copy of the DNA that's going to be carried out into the nucleus. So it literally carries the message of DNA. Ribosomal RNA, also called rRNA, is the molecule that makes up the actual physical structure of the ribosomes. Since ribosomes have to make protein, they can't be made of protein. They're made of nucleic acids. And the last one we're going to focus in on a little bit more when we get into the second part called translation. This is called transfer RNA or tRNA. I'm going to save the function of that for a little bit later. But basically what it does is it carries those building blocks of proteins called amino acids to the proper message on the mRNA. So we'll take a look at those in more detail later. Now, mRNA is that portion that's going to carry that gene that's going to make the protein out of the nucleus, and then it's going to take it to the site of protein synthesis, that being the ribosomes. So that's a little summary of that. Ribosomal RNA is basically making up the ribosomes. You don't have to worry about the 2 subunits, but I want you to think of a ribosome as a hamburger bun. It's got a top unit and a bottom unit. We're not going to focus in on a lot of these different structures here. Just know that rRNA is what makes up the ribosomes. And then, as I was saying before, tRNA or transfer RNA, that's what's going to carry those building blocks or amino acids up to that message on the mRNA. Now, the first process is called transcription. To transcribe something, you're literally just rewriting it. So we're taking DNA code and we're writing RNA code. But the way that we do that is we add bases in a complementary fashion. So what that means is when we're reading the DNA strand, this RNA strand is actually going to be sort of a mirror image of what the DNA states. So if you take a look here, there's an enzyme involved. That enzyme is called RNA polymerase. And what it does, you can see one of the parts right here is that it unzips the DNA. But the other part is that it adds complementary base pairs, but it's going to add RNA nucleotides. So if you take a look here, this is a red base on the DNA. That's guanine. Opposite of that is cytosine. Here you can see thymine. Opposite of that is going to be adenine. Now this green one is adenine. Normally you would expect to see thymine opposite of that, but now you can see it's orange. Here is where you're going to use that uracil. So what it's going to do is it's going to build a segment of RNA from the portion of DNA that it reads. So this circle is representing the active gene that we're reading. And that's the RNA molecule that's being built from it. So, what does RNA polymerase do? It only binds to those areas of the DNA that are actually going to say something. There's a lot of areas of DNA that we don't want to translate or transcribe because we don't need those proteins at the moment. So we have regions on DNA called promoters. And basically what they have is a specific sequence of bases that say this is where an active gene that you may want to translate begins. So the key term here, promoter, is a region of DNA that starts the transcription process. Once the RNA is made, we're going to trim out some of the garbage. Now, the first thing that we have to do is we have to take a look here. These are called introns. Introns, I always think of them as they code in between the active portions. The other portions in dark blue are called exons. So what we want to do is get rid of these introns, and we want to splice together what's left. Notice that the final RNA molecule is much smaller than the original. This is called RNA editing or RNA processing. We also add a cap and a tail on them because there are some nasty things out in the cytoplasm. Those are enzymes that are meant to break these things down and recycle some of those free nucleotides for use later. We want to make sure that we don't chew this molecule up too fast. So we add a cap and a tail to protect the stuff on the inside while we're processing it. So as you can see there, those smaller circles, those are the exons that we want to keep. Okay, so that's a real fast crash course in RNA synthesis. What we're going to take a look at in the next video is the actual process of making a protein. And after that activity, I'm actually going to try and give you guys something that's a little more basic. It'll give you some initial feedback to tell you very quickly if you're doing it right. It's one of those things you can do it and you figure out real quick if you're doing it right or wrong. So I'm going to save that as a surprise, and I'll tell you about that in the next video. So I want you to try that interactivity related to this exercise and see if you have any questions on that. Write those down and bring those to our discussion during office hours. Okay, so good luck, stay safe,